so I think we have pretty much everyone here. Are we missing? Who are we missing? Actually, we're missing a couple of grade 11 students. They're not here yet? All right, Audrey's not here. There's still only, they're missing 4 of you. Okay, so I think we actually, this is everybody that is going to join this morning. So some of them are missing other classes. Maybe we can just introduce ourselves really briefly, and so there's students from grade 10, there's students from grade 11, and there's students from grade 8 here.
Awesome.
Yeah, so this is high school. Correct me if I'm wrong. Tyngsborough, right? Is that how you pronounce it?
Yes. Yes.
Yeah, so Tyngsborough in Massachusetts, and this is their BioBuilders group. And this is Miss Rebecca.
Yep. Hi.
Can you see okay? Yeah, guys, if you want, the camera's over here. I don't know if they can.
All right. That's a lot, yeah. So, do you want us to introduce ourselves individually?
Sure. Yeah, that'd be great.
So, I'm Miss Rebecca. I'm the advisor for the Tyngsborough BioBuilder Club, and next is.
I am the head of fundraising for BioBuilder Club and Robotics at Degrading School.
And you are what grade?
I'm a senior.
Excellent. Next.
I am Trey. I am also a senior, but I am the president of the robotics team.
Oh, nice. Wow.
I'm Jackson, and I'm a senior, and I am captain of the tennis team.
Oh, wow.
Next. Don't.
I'm Joey. I'm in 9th grade, and this is my first year of BioBuilder.
Hey!
Hi, I'm daughter.
Oh, you're forced to be for you. Okay.
My name is Colin, also freshman. BioBuilder.
Yay! All right, so
I'm Sophie, I'm a sophomore, and I'm in BioBuilder.
I'm
I'm a sophomore, this is my second year.
Yay. Now
I'm Kate. I'm a senior also, and I'm the one that's been emailing for the past few weeks. And yeah, I traveled to Indonesia this past summer and lived in Jakarta for like 6 weeks.
Yeah. Hi, Kate. How are you? Yeah, we've been in email correspondence, right?
Yes.
Yeah. Selamat pagi. All right. Yeah. So Kate actually went to Indonesia this summer.
all right. Do you introduce everybody there?
Sure.
Or is that anything?
Okay, so maybe a quick introduction. There's, I think there's 20 people here, so maybe you can just stand up very quickly and introduce yourself. Yeah?
Yeah.
So go ahead, we'll start with Kyra. Kyra?
I'm Kyra. I'm in grade 10, and it's my first year.
Yay.
Yeah, very good. Rania? Yeah, we'll start.
Hi, I'm Rania. I'm in 10th grade as well, and it's also my first year here.
You're going to have to get
Excellent.
Sorry, if we turn on the lights here, it's hard for us to see you. So we're going to turn the lights off when you guys start, but for right now we'll leave them on so you can hopefully see the students.
Okay.
We have Trey.
Hi, my name is Yen, and I'm the vice president of the
BioBuilders.
BioBuilders Club.
Hi, I'm Mateo. I'm new.
Can you hear okay?
Yes.
Okay, good. Hey, come on, guys.
Hi, I'm Sherry, and I'm in grade 8, and this is my first year. Hi, my name is Evelyn. I'm also from grade 8, and this is my first year.
Great.
Hi, I'm Gisela, and I'm also from grade 8, and this is also my first year.
Awesome.
Hello, my name's Gary, and I'm also in grade 8, and this is also my first year.
Okay.
Hi, my name is Jordan. I'm also in grade 8, and this is also my first year.
Hi, my name is Kay, and I'm in grade 8, and this is my first year as well.
That's good.
Maybe something else aside from this is your first year.
Hi, my name is Hannah, and I'm in grade 8, and this is also my first year.
Okay.
Hi, I'm Tushar, and I successfully graduated grade 9, so I'm in grade 10 now. By the way, this is my second year. Nice to meet you.
I am Matthew J, and I'm in grade 8. This is my first year in BioBuilders.
Awesome.
Hi, my name is Jungwon. I'm secretary for Bio2M, and this is my second year joining Bio2M.
Hi, my name is Chris, and I'm in 10th grade.
Very good.
Hi, my name is Ishan, and I'm also in 10th grade.
Yeah, so that's everybody that's here. Here we do have a BioBuilders group, but we call it B2M because they do actually a couple of other things. So the BioBuilders part is kind of like a part. Many of the students here are interested in medicine, so they're, we do like dissections and things like that. So
That's great. All right, awesome. So I have a slideshow that goes with the BioBuilder lab. If you want to watch that, would that be
yeah,
good?
that'd be great. Yeah.
Okay, awesome. So I'm gonna go to the, let's see, how do I go to present? So I hope this
I think it's a
works.
yeah. At the bottom.
There we go. All right. Okay. Can we see that?
Yeah.
All right. So, quiet back here. All right. So I'm so delighted that we're able to chat with you guys today. This slideshow was developed and put together by Natalie Coldell, who is the founder of BioBuilder. And so this is a slideshow that she has produced mostly for teachers, but I feel like it's a nice compilation of what teachers and students can experience from BioBuilder itself. And if you have any questions as I go through this, please feel free to interrupt. And that goes for my students as well. Right? Yes? All right. So at all of the BioBuilder labs that are offered, sort of move students and teachers through the design, build, and test cycle. And so this lab in particular, What a Colorful World, is going to look at the building component of it. And as Chris mentioned, it is essentially a bacterial transformation lab with a little bit of a twist. And so it will be interesting to see how that connects with this whole sort of broader topic of synthetic biology. So just to kind of introduce the lab itself, the idea behind the lab is to introduce students and teachers as you're sort of designing your projects to be thinking not just about what the parts are that you're going to use to build your genetic program, but to also consider what the chassis is. So what kind of cell type are you going to be using, and what kind of cell type do you want to run your genetic program in? So much like there are a lot of different chassis that we can find with cars, car makes and models vary from the Studebaker to the Lamborghini to the DeLorean right in the middle. They all have different features that are attractive to different prospective drivers and car owners. In the same way that cars have chassis that you can sort of build features into, cells also have these variations. So they vary in size and shape, the number of organelles they have, the kind of cell that you can deal with, whether it's prokaryote or eukaryote or some kind of virus as well. Picking the best host to reprogram and to run your genetic program is a really important part of the design process. And you can either start with that. It sounds like you have some experience in this, so you can either select your chassis beforehand and then design your program around those features, or you can do it in the opposite way. And that totally depends on what your focus is. So this activity, this lab, What a Colorful World, is looking at running two identical genetic programs into two different strains of bacteria. And so we're gonna run these programs in a strain of bacteria that we call 4-1 and also 4-2, which I believe are analogous to K through 12 E. coli and then the B strain of E. coli. The programs that you're going to run have already been, or that we're going to demonstrate in the transformation here, have already been pre-programmed. So it's not as though we're sort of building and assembling these pieces of DNA. These parts are already in the DNA that I will distribute to the students here. And they look like this. So if you can recognize, some of these parts are sort of recognizable here. The arrow obviously is a promoter, and these are constitutive promoters. And then the sort of green shape and then the purple shape are indicating the genetic information that codes for the expression of a green pigment and a purple pigment. So schematically, what we're going to be doing is the students will be transforming both the purple plasmid and the green plasmid. So these color generating devices, they're going to transform into two different strains of E. coli, and that's what's being illustrated here. So just like many of the other labs that BioBuilder offers, a lot of them are inspired by past iGEM projects. And I don't know if you're familiar with iGEM. It sounds like obviously you have some experience here, so you probably are familiar with the International Genetically Engineered Machine Competition. And it's a really great resource. So if you go online and you're trying to find projects and find sort of, places where you can be inspired and maybe even continue on potential projects, this is a really great place to kind of start. The 2009 iGEM team from the University of Cambridge wanted to devise E. coli to produce different types of pigments. And the purpose for doing that, I think, if you go to their wiki page and look at their project and their poster and also their presentation, which is all sort of curated by the iGEM organization, the idea was to be able to produce pigments in E. coli as opposed to producing an output that was like fluorescence or a scent that was really difficult to be able to evaluate as an output. So they wanted something more visual. And so they named this E. chromide. There's a lot of talk behind me because we also have a middle school information night going on right now. So I apologize for that. So again, if we look at the diagram here, essentially what the University of Cambridge wanted to do is to build these living biosensors for the detection of specific environmental contaminants like heavy metals like arsenic and mercury. They didn't just want to detect their presence with a color output, but they also wanted the pattern of color to provide information about the concentration of those biological contaminants. And so they wanted it to be more visual, and they wanted to be able to make determination about the concentration and content of a water sample as a result. So it turns out that the first place they went to was nature to see what kind of colors were naturally produced by different bacterial sources. The bacterium Chromobacterium violacine is a soil bacterium that they discovered that showed great possibilities in terms of getting their pigments to their project to move forward. And when they looked more at sort of the biochemical pathways for the production of different pigments, they noticed that this particular bacterium begins with tryptophan and requires a sequence of 5 different enzymes that are used together in order to generate a purple pigment. But so the enzymes are listed here, A, B, C, D, and E, but they are not used in that order. And so they noticed that when they removed BioC, or that C enzyme from the biological that pathway, they were able to produce a green pigment as well. So with one biochemical pathway, one genetic pathway, they were able to produce 2 different color variants. And so this was the inspiration for What a Colorful World. I don't know if you guys do this, but this is also a really neat opportunity to maybe talk about logic gates and truth tables. And so when you design your own systems, your own genetic programs, you can maybe describe them in terms of these methods. So if we use the What a Colorful World those genetic programs to describe them in terms of logic gates, you'll see that we have this little AND gate up at the top left here. And if we run program X and Y, we get output Z. The output Z should be a purple pigment. If we remove input Y and we just run the X program here, what we're going to end up getting is a green pigment. If you have neither of these, or if you're just running an input Y, then you're not going to get any pigments as a result. So what we're going to do is perform experimentally the bacterial transformation. We are going to make our cells chemically competent by using calcium chloride. That calcium chloride is able to sort of charge up that negatively charged membrane with a positive charge to attract those plasmids of DNA that are negative. We're going to expose our system to a little bit of heat to make our cells a little bit porous so that the DNA will be able to be taken up by the cells, and then we'll seal up that cell membrane, plate them on some LB amp plates, and then hopefully we'll have a result by tomorrow. And this is the transformation protocol. I'm actually gonna just roll through this very quickly because I'm gonna actually have my students just share the experience with you right now. So I'm gonna get out of the slide here. And do you have any, I'll stop presenting here. Do you have any questions about the lab itself?
I think, can I just give them a minute to discuss with each other?
Yeah, sure.
Okay. So yeah, guys, if you can discuss this with each other for, let's say, a minute or two, right? And then if there are any questions, we'll ask maybe each table to ask a question. Okay, so.
It's hard to
Okay, so you guys start with a question. So stand up, please. All right, where's the question?
Oh, okay.
All right. So you're gonna have to come here. Sorry.
You guys are in there.
You gotta go put your head down a little bit.
Bye.
I actually had a question about like what kind of materials does it take to conduct such experiments?
Oh, so you mean like the actual equipment materials? Yeah, that's a great question. So the first thing that we're going to need are some agar plates, right? Can I just see? So these are the agar plates that have an LB, like a nutrient, so it's like for the bacteria, but it also has the antibiotic ampicillin. So the programs that we're going to be running today are ampicillin resistant. So only the cells that have taken up our DNA will be able to grow on these dishes. Okay. There are a lot of other materials here too. So I don't know if we want to go through all of these. But what I'm going to do with my computer is set you up right next to a group so that, and as they go through the process, they'll show you what the piece of equipment is and how to use it. Okay. Would that be helpful?
Yeah. Yeah.
Okay, great.
Thank you.
Just two more, sorry.
Hi, it's nice to meet you. My table was wondering how many times you reprogram your program to make it like actually work.
Every time. Oh, like for this lab?
Yeah.
This lab works all the time. Our actual projects never work.
Oh, okay. Thank you.
You're welcome.
So there was a question about is it always successful?
Yes, this lab is always successful.
Okay.
Yeah.
Okay. I think
I mean, unless of course you're overheating the cells and then you kill them in the process, or you don't pick up enough DNA. So there's a lot of places where error can happen, but I think the concentration of DNA is like perfect. It's optimum, so we always get results.
Okay, awesome. Thank you. Okay, yeah, I think, we're ready to continue.
We're ready?
Yeah.
Okay, great. I'm gonna put you next to this group. They've done this lab several times, so they shouldn't
nice.
mess up. Right, guys? Right. Okay. All right, so here we go. Actually, you know what? Can I do this instead? You guys. It's sealed. Okay, we're gonna do that. Okay. There we go. All right. Can we see? Does this work? Yeah?
Yeah, we
Does
can
that
see.
look good to you? Yes?
Yep.
Okay. Great. All right. So these guys are gonna help you out. This is Kate and Trey.
Hi, Kate. Hi, Trey.
Hi.
Okay.
Follow the aprons, and there's no way to left, right? Because you're handling bacteria. So calcium chloride. Right there. Calcium chloride.
Okay.
Okay.
Yeah. So, we have goggles and gloves on for safety. And then we have our directions all labeled out right here. And they're laminated, so if anything spills on them, nothing will be ruined. And yeah, basically we just follow the directions step by step, and we'll walk you guys through it. Right.
Okay. So first we're going to have to label these two microfuge tubes. We're going to have to label one 4-1 and label the other one 4-2. And that's to represent the two different strains we're using in this experiment.
Okay.
Yeah, we're just using a Sharpie. Right.
Okay, so can you take notes, please? Keywords that you hear, right? Try and outline the process.
So, just like this. You can talk to it.
Alright, and then next we're going to pipette 200 microliters of the calcium chloride solution into a microfuge tube, and we're going to use that using a micropipette.
A micropipette, calcium chloride, 200 microliters.
200 microliters.
So we have our calcium chloride right here in the small centrifuge or micro tube thing. And then Trey's gonna use these micropipettes, and they're kind of complicated to use at first. It's just like a fancier version, a really exact version of a regular pipette. But the way that they work is that you turn the dial until it gets to the number that you're supposed to go to, and then you put the tip on, which all the tips come in these little boxes. So you can't use the same tip twice on different substances because then it would be cross-contamination. And you stick the tube in here and press the button, and then you're able to do just like Trey did.
And then afterwards, you just release the tip into a bleach solution.
Yeah, so then no chemicals.
So the bleach is going to kill the bacteria, anything that's left on the tip right.
Okay, after this, we're just going to place the tubes. We're just going to place them on crushed ice, cool down.
So there's calcium chloride in there, right? You put it in ice, use the micropipette.
So now that each of the labeled tubes are on the ice, so they're going to be cooling on here, and there's the same thing in each one, but there will be different things in each one later, and that's why we've labeled them each one differently.
Okay, next we're going to take cells from each respective strain, and we're going to put them into the corresponding tube.
So the cells, the bacterial cells are in those petri tubes.
So this is an LB tray with actual cells on it. So we have them split between two sides, and there's the 4-1 strain and the 4-2 strain right on here. And we use these two, what are they?
Inoculating loops.
Yeah, these inoculating loops. And they come in bags, so they're sterile, and you can't touch them. And then we use these to get the DNA on here, like a group of DNA, and then this is how we'll mix it into the.
Or one's closer to you. Alright, so first we're going to do the 4-1 strain. Yeah, you just lightly scrape. Yeah. Okay, that's good. And
So then I scrape them directly onto this little circular thing.
then what we're going to do is we're just going to place them into the 4-1 tube since we took from the 4-1 strain, and you're just going to put them in and swirl to release the cells into that solution.
Yeah, and you want to make sure they're all off. Okay.
Yeah. And afterwards, we're just going to dispose of this since now it has cellular material in it, and we're just going to put it in bleach. And then we're also going to put it back on ice to cool it down. And then we're going to take the 4-2 strain and repeat the same process. 4-2 near you.
Yeah, we did have a new one, right? And then a couple of times. It's all set. You have enough stuff.
The DNA actually smell kind of weird.
Yeah, so the cells that are, so, the reason why you want all the cells is because you want to make sure that you're.
So you can see this off of DNA on this one.
Adding cells to that solution, right? Is that a little?
And we're going to place it back on ice like we did the last one. Right. Okay. And next, in order to mix them together, since we don't have a vortex machine, which is this little machine we put it in and spins the cells around and mixes it, we're just going to mix them by flicking them. So if you just gently flick them, then they'll get mixed with the solution, the cells. Alright, then we're just going to return them after they're mixed. And then afterwards,
What's an aliquot?
Yeah, what is that?
Amount.
What is that?
So I took, are we talking about the DNA?
Yeah.
Yeah, so I received like 60 microliters or something. I aliquoted, I gave each team 5 microliter samples.
So we're just putting
So we already have a specifically measured out amount of each of these 2 substances, and our teacher kindly put them in different colored tubes so we know which one is which for the lab. And this is the plasmid that we're going to be adding.
So those are the plasmids, that's the DNA, right, that they're gonna add to the cells, or to the solution that has the cells for transformation.
And we'll do the same thing for the rate.
next we're just labeling 1/4. Okay, so next we're just gonna label, so for this, for the purple plasmid, yeah, for the purple, we're gonna label one the 4-1 strain, and then the other the 4-2 strain, and then we're gonna repeat that for the green plasmid as well.
The 4-1 strain, and then what you're gonna do is type 100 microliters. So these 2 cells with the cells in them. So you just want to put these to, what are you, 4-2?
Okay, so next, we're gonna change the micropipette to be equivalent to 100 microliters. So you just do that by, if you spin the knob at the top, some micropipettes are different on how you change the amount. We're just, well, you can spin it. It's fine. Alright, so now it's set to 100, and we're going to take 100 microliters of the we're just gonna start the 4-1 strain, and we're gonna put 100 into one green tube and then 100 into a purple tube. So the 4-1.
Change out your tip. Make sure you guys change out your.
So it's helpful to have them both open, just so it's easier to transfer it.
Yeah.
Except.
And then we're going to dispose of it since it went into something containing the plasmids. We don't want cross-contamination to occur.
Feel free to stop us if you guys have any questions along the way.
Any questions?
Purple 4-1. The 4-1 is green.
And it's such a little amount that you can almost barely see it, but we know that it's exact from using the micropipette.
Alright, next, we're going to repeat that process, but we're going to use the 4-2 strain now.
So, can we ask a question? What's the difference between the 4-1 and 4-2 strains?
The 4-1 is the K12 strain, and then the 4-2 is the B strain. There are some metabolic differences.
Okay.
If you're scraping up the cells to 4-2 clumpy. So
Okay.
do you have a BioBuilder textbook?
We do,
Did you
yeah.
get that?
We do. We have it,
So
yeah.
there's a chapter dedicated to this, and it will
Okay.
describe to you some of those characteristics too.
Okay.
Yeah, which will influence the expression.
Okay, thanks.
You're welcome. Right. Now, remember the cells that you have here, right? So like this, right? So you would put out your. There's nothing to take 100 micrograms.
What?
Don't trust me. There's stuff in there.
Cells.
There's stuff. Okay. Next, we're just going to inoculate these on ice for 5 minutes. Okay, we're just going to cool them on ice like that. I was reading the procedure. Alright, so now we're just going to wait 5 minutes. We're going to let them cool down. And I'm just going to set a timer. It doesn't matter if it's exact, it just needs to be around 5 minutes because the amount of time you're cooling it, you just want to make sure it's at least 5 minutes. So now we're just going to sit back and wait.
Do you guys have any questions about while we're waiting? So who can summarize what they've done now up to this point?
Sure.
So guys, who can summarize what they've done up until this point, right? They're transforming bacteria.
Oh, right. Yeah.
Well,
you've already done 5 minutes of ice.
No, you don't have to.
Right, how much time do you have?
You have 41 seconds in.
Pretty much. Yeah.
Yeah. Oh, whoa.
And everybody just.
So after the 5 minutes, the next step will be to heat shock these two. And this is the machine that we're going to be using to heat shock them. There's actual little container areas that we'll be able to put them in, and then we have a thermometer in the middle to measure the temperature. The actual temperature is 42 degrees Celsius for 90 seconds exactly. And the heat shock part actually has to be a little bit more specific than when we put it on ice.
It's too much. Alright.
So what do you guys think the heat shock is going to do? So these bacteria are cold, calcium chloride, and there's DNA flowing around them.
Wait, we need to put them right there.
Yeah.
The temperature, they're in ice right now, right? So they've been just chilled. There's calcium chloride all in the solution, and there's plasmids. And we're going to put them in the solution. The temperature's going to shoot up for a while. And hopefully some of them will take up the plasmids.
Yeah, I know, yeah. So it is on, it was unplugged, and so it's not at the temperature we need it at. This might take a little while. No, it's outside, it's heating right now, which is, so it's gonna be a little bit longer than we expected. Yeah, it'll go up, I mean, it'll go up pretty quickly, but, yeah, I mean, I don't know what the.
Like 2.5 point.
You got 2.5.
Yeah, it takes like 3 Mississippi.
Yeah.
The water bath, there's little spaces for
It
micro
takes like 2 seconds per thing.
But, it sounds like it wasn't.
I mean, like it'll go up 3 degrees.
But I think the closer that you get to the slower it's gonna.
I don't think that's okay. I think it'll go faster.
Oh, like it's.
You know, yeah, make it slower.
Do you need, how many do you know?
4.
I like the exponential.
Yeah.
It will increase. See, look at it zooming now.
So while we're waiting,
Basically, we're just waiting for the temperature to go up.
you can summarize what they've done.
Maybe you could talk about maybe your project and their project, like what are they thinking about in terms of.
Yeah, Kate, you got
That's
that.
a great idea, okay?
Kate, you got it.
They're gonna have.
For a winner, a potato, like smash it all up.
I would expect.
Alright, so have you guys thought of any ideas for your project yet this year?
They're still saying.
They're in the process. They're in the process of working on these different projects. Last year, our project was on cells that make insulin. So the group, most of them are in grade 12, and unfortunately, they can't be here this morning. But the idea was that they have, E. coli that can respond to like sugars in the large intestine. And then in response, they would produce insulin to combat kind of like a natural insulin delivery system. The time before that, or the year before, we did a Tmao degrading system. So Tmao is this compound that I guess kicks off the initial events of atherosclerosis, and it's found in fish and meats and things like that. And so yeah, we had another kind of probiotic system where it was like something that would be living inside of the person and then allow them basically to eat meat and fish without getting this Tmao. So yeah, but please share with us. They're still kind of in the very early stages. Yeah, do you want to share ideas or things that you guys are working on?
Yeah, Kate can share.
We're continuing work with our Alive Light project that we started last year, which is basically just a natural light generating machine, and we're using like a combination of yeast and the protein luciferase, and it ends up glowing. So we had our design brief published from last year, and now we're working on continuing that project, and hopefully we're going to be doing some actual lab things rather than just research. And yeah, we're also working on some like marketing aspects of the project too.
Oh, cool. Like what do you mean? Marketing aspects. What did you mean?
Well, since it's like an actual light, we were talking about how we could glowing different colors, or even having like a scent for different like holidays and stuff throughout the year. And we also talked about incorporating something to make it also have like a bug repellent, like a mosquito repellent type of thing that the light would include. So we're kind of developing those kind of progressions with our project.
Oh, that's very cool.
Tell them about citronella and mosquitoes.
Oh, they said that.
Yeah,
Talk about
No.
I don't know. I'll say it wrong.
Yeah.
Oh, Celsius.
All right, so we're just waiting for the heat block. I apologize.
Share with them ideas about your project. I think Kyra, Kyra, I know you have. Do you want to come up and tell them about your project?
Oh, yeah, let's do that.
Okay, come explain it to them in
What do we
brief.
want to hear?
The
Your personal project,
the
yeah.
PP project.
Yeah.
Oh my god. So basically, there's like this enzyme called peptase, and it's found in this bacteria called Ideonella sakaiensis. And what this enzyme does, it breaks down the PET plastic. So I was thinking like if you're able to make a plasmid with that enzyme, and you can put it into cyanobacteria which live in the water, then it would be able to degrade the microplastics in the oceans. Yeah.
Oh, that's cool.
Yeah, that's super.
That's great.
Plastic.
Yeah. Plastic's a good. That's a good project.
Yeah.
Yeah.
I feel like it's time to implement now.
You think so?
Yeah.
I mean, like, will they die?
They won't die, but it's not 42 degrees. So
42.
I'm looking at the thermometer from here, right?
Oh, boy, they're at one.
Oh, we haven't been through that.
Usually, more and more in the bathroom.
We have another student that would like to share with you. Sorry.
So my project was about detecting a protein called cardiac troponin T in the bloodstream that and the higher the concentration, the more chances the person will suffer from myocardial infarction. So basically, what I wanted to do is use bacteria that produces a protein that binds exactly to the other protein, and when this happens, there'll be a change in current, and that change in current can be detected using a really sensitive ammeter, and that'll correspond to the change in to the concentration level.
Wow, that's awesome. That is very cool. Have you guys gotten to any build stages yet?
No, we're all totally theoretical at this point.
Yeah, that's awesome. So the PET project that your student was talking about, if you go to the iGEM website, there are some past iGEM projects that have dealt with PET-A's and MET-A's and that whole pathway. So that might give you some idea of where to go with it.
Yeah, I was looking at like the Yale website, but then it was very I was still researching.
Yeah, that's awesome. That's great.
It's falling below.
I don't know what else to say.
Okay. 5 degrees away.
It's falling.
Oh, because you keep transferring this into this. But you can get started that again if you don't know.
You guys have done us for training. Yeah. How often does your BioBuilder club meet?
Like once a week.
Yeah.
Do you do it after school or during school?
After school.
After school. Cool. Is it for a couple of hours?
1.5 hours. Yeah.
Oh, that's great. Wow.
Yeah, kind of the biggest problem that we have here is access to DNA and enzymes. So Indonesia doesn't really have like an enzyme industry, so it's all imported. And the same with like, if you want to make DNA or, just have it, a primer or something like that, it's all from Korea. So it's starting, but it's still, that's the hardest problem, right, is to get, those kind of things because they're all imported. There's no local industry for that.
That must be very expensive too.
Yeah, well, we haven't really done it yet. Yeah. So there's a couple of universities that we've worked with, and they kind of charge us a fee to go and use their like facilities. It's pretty reasonable, but most of them are actually kind of far away. So we're in a suburb of Jakarta, and the closest one is like an hour and a half away, which makes it hard to, if we were going to do it a couple times. Yeah.
Wow.
Yeah.
Wow.
Yeah,
I wish we could send you all of this stuff.
Well, we have some cells that we took back from one of our university trips, and we culture them here. But the plasmids and enzymes and things like that, those are more difficult. So, maybe I'm gonna come home for December, so maybe I'll get some stuff and bring it back.
Oh yeah, that'd be great.
It just takes time and money. So like we ordered a bacterial transformation kit like 10 years ago, and it costs the duty is more expensive than the kit. So it was like $150, so we had to pay $200 in tax.
I think we should go for it.
How long have you been in
For a long time. All right, guys. I think we're ready to heat shock these little cells.
Awesome.
Okay.
So
All
they're
right. Let's shock the cells.
Okay.
All right. You make sure you do it on time. Yeah, let's do it on time, guys. So bring all of your
Okay.
your green.
So now we're just gonna heat shock the.
90 seconds. The timer.
I have it ready.
Wait, everybody.
Yeah, all of it. Go. Go.
Wait, don't.
Down 30 degrees.
10 seconds.
When they're done.
20 seconds. No, they can just do, when we get to 20, we can just and then end it when we end at 110.
20.
Never good.
Okay. And after this, we're just gonna move them to room temperature to let them cool down a little bit.
Yeah.
40.
Once you're done, then make sure you go back and label your plates. Okay, so your LB amp plates, you should label these purple 4-1, purple 4-2, green 4-1.
Ooh.
So,
So we're done.
Let's pick it up. Oh.
Huh?
You screw it in there in that hole.
Our
70 seconds.
excellent.
Well, you need to plug this in.
15 left.
You should try that.
T minus 15. T minus 10. T original. T original. 5, 4, 3, 2, 1.
They can only grow. No, sorry, they'll be just
Okay,
Now we're gonna do it.
Okay, you guys can time yourselves. Okay, so next we're just going to add we're going to add 0.5 milliliters of LB to each cell, each container, and we're just going to use that using just your normal disposable pipette. You get the exact measurement, doesn't matter since it's just the food that for the cells here. And Kate can do that. Wow. 0.5. Yeah. So we're just going to do that, and then while she is doing that, I'm going to label each agar plate, and we're going to label one 4-1 purple, one 4-1 green, another 4-2 purple, another 4-2 green. So we're just going to label. So we're just going to label it similar to that. I don't know if you can even see that.
Yeah, we
Yes.
Sure. You can change out your pipette tips, okay? Find that you're adding LP into your sample and then mix, right?
Sorry, Kate. There was a question from a couple of students. They wanted to know what grade you are all in.
We're both seniors here.
We're both in grade 12.
Okay, grade 12.
The two other boys right here are both in grade 12. These three in the back are in grade 9. And then these two girls over here are in grade 10.
Okay, thanks. Yeah, so grade 9 to 12 guys. Yeah, most of the people in the front are 12. I mean, like, how long have they been there? Sorry, I don't want to interrupt you during the protocol, but there were questions about how long you've been studying BioBuilders, like in general, and then how many times a week do you devote to your BioBuilder project?
Yeah, of course. So our group pretty much. Our group pretty much started last year. Last year was our first year. Our teacher, Mrs. Rav, had a BioBuilder club before when we were in high school, but it died off, and then last year we started it up again because my grade. So me, Trey, KJ over there, and Jackson were in AP biology class with Mrs. Rav when we were in grade 10, and then we started the club the year after, and other kids joined. So we also were in freshman year. In grade 9, we had a biology class with Mrs. Rav. So all of us took biology freshman year and AP biology sophomore year, and then we've been doing BioBuilder club since then. And then our group meets every single Friday for about 2 hours.
Oh wow. Okay. Thank you.
Yeah.
Okay, so next what we're going to do is we're going to gently just invert it to mix all the stuff we put in together. And then what we're going to do is we're going to take, so this is 4-1 green. So we're just going to find the corresponding agar plate, and we're going to add 250 microliters of this sample into the appropriate petri dish. So what we're going to do is first we're just going to take this. We're going to use a micropipette, but this time one adjusted to larger quantities.
Alright, so the spreader.
Alright, so we're just going to put it on there. And then what we're going to do after that is we're going to take one of these inoculating loops, or actually we have spreaders. So you can use an inoculating loop, but it's also easier to just use the spreader if you have those available to you. So this is the spreader, it's just basically just a piece of plastic. What we're going to do is we're just going to evenly distribute it. So we're just going to spread it around and first in a circle, and then we're just going to make sure everything is covered. Alright, so now it's spread. We're just going to put the top back on, and we're just going to let it be. And then we're just going to repeat that process for each one. So the 4-1 green is on. So now 4-2 green. So now we're going to take 4-2 green and repeat that process.
Were you guys able to do the online lab simulations that the BioBuilder is talking about in the next online meeting?
We've played around with a couple of I don't know if it's that exact one, but they're on PHET, and you can see the effect of binding strength and the amount of DNA.
Yeah, I know I'm not sure, I have to ask Ms. Rav if there's an iGEM one, but they walk through kind of this lab. Ms. Rav.
We did it in not this year, but we've done it in the past. It's pretty cool, right? Yeah, it's from the bento box.
Yes. Oh, what is it?
Yeah, they said they did it. They said they did it last year.
Yeah, they did it last year, but
Yeah, I don't remember that. That's pretty good though. I think it's a pretty good representation of this.
You want to start that one? No. You want to spread
I
that?
haven't learned that. Those two problems.
Can you spread that?
The ones that were really long, the
Yeah. Yeah, negative x squared. Minus negative x squared. And it's equal to x squared.
I don't know how to do it either. I spent so long.
They'll know that
Sorry guys, we're asking them to do a summary of everything that they watched here, and it's just kind of hitting them, and they were like, oh my gosh.
Okay, so now all the cells have been distributed on each plate in coordination with what that cell is contained. So now we're just going to incubate the petri dishes overnight, and that will help the cells grow, and then we should see our results later that same day.
And these ones where we Fred. And these ones where we see under UV light. And these the ones that we see under UV light. Okay, so probably if we're able to look at these cells tomorrow, we can send you guys images of them all.
Oh, that'd be awesome. Yeah.
Yeah, of course. We also will be able to look at the. Is this the green protein? Yeah. We'll be able to look at the green fluorescent protein underneath the UV light, and we'll be able to see the cells really well with that. I'm pretty sure.
It might not be. It's green something. Basically, it turns green under something. Is it GFP? No, it's
the ones
not.
that we look at under the UV light? No, these aren't.
Oh, we lied. They're not under UV light. They just.
But we will be able to send you guys pictures of them tomorrow.
Okay, that's awesome. Thank you so much for letting us watch.
Yeah, of course.
You're welcome. So I'll send pictures of what the results are.
Wait, can you all stand up maybe and come closer so I can see? You know,
They're out.
Thank you.
Thank you.
Alright.
Thank you.
ありがとうございます。
Okay, so thank you. Sorry it was a little bit later than we thought. I made a screencast of this, so I'll share the screencast with you by Google Drive. Okay?